Light is everywhere around us, helping us see and experience the world. But what if I told you that this light can help us understand our inner world better? That's our biology. Don't believe me? Well, this is exactly what optogenetics does. Through gene editing, we can insert light sensitive genes into biological cells so that we can control their chemical activity through light. The best part is that this is reversible. When light stops shining, the chemical response goes back to its initial state. One problem I wanted to solve with optogenetics was the off-target effects of CRISPR-Cas9, in which wrong genes are targeted for insertion or deletion. Yes, you heard it right. Using a technology that primarily used gene editing to make gene editing more accurate. I wrote a review paper on Opto CRISPR-Cas9 tools, and here's what I learned. CRISPR-Cas9, which locates and cuts the targeted genes, has two main functional components, single guide RNA and Cas9. Single guide RNA is complementary to the target DNA. Once it's released into the cell, it finds its complementary pair of DNA, and Cas9, which is a pair of molecular scissors, cuts it. The problem comes when CRISPR-Cas9 doesn't accidentally locates the wrong genes, leading to off-target effects which can even lead to cancer. To make it more accurate, we can make CRISPR-Cas9 light sensitive. Why? Well, because light is non-invasive and spatiotemporally precise, which means we can control the time and space where gene editing occurs. It's also non-toxic in most cases and reversible. To make CRISPR-Cas9 light sensitive, we can't use gene editing because it is the tool that does gene editing and it's a molecular system. So we dive into the tools of molecular biology to solve this problem. One system that we can use is called split systems or light-induced dimerizations, in which one molecule is broken into two parts and attached to light-sensitive domains, which are molecules that respond to light. This component in this state is non-functional, but when light is shown, the light-sensitive domains recombine and rebuild the original component, making it functional with the switch of a light. This has potential in anti-cancer therapies because it can control the time and space where gene editing occurs through the controlled exposure of light. Often, great discoveries are made when two diverse fields combine to extend their capabilities. Optogenetics was originally developed to control neurons, and now it has joined forces with CRISPR-Cas9, the most versatile tool for gene editing to exist yet, paving the way for new discoveries and treatments. What new discoveries can be made by intersecting more interesting fields?